Well, again, good evening, everyone, and welcome to the AMS monthly symposium. I'm Jonathan Plett. I am the president of the society, and it is my pleasure to welcome tonight our speaker, who is going to be Marc Freestone. He has worked for a number of years in the conservation space. His specialty is in the area of orchid mycorrhiza. When he was here in Australia, he was looking at the distribution of orchid mycorrhiza, culturing and using those mycorrhiza as a means of maintaining and preserving orchids here. He's also was a regular contributor during that time to ABC, sharing his knowledge within citizen science. He was supporting orchid conservation programs and worked also at the Royal Botanic Gardens in Victoria. So with that, I'm going to pass it over to Marc. If you want to start sharing your screen, Marc, and we look forward to what you have to talk to us about tonight.
Thanks, Jonathan. Can you hear me all right there? Excellent. Um, well, yeah, good evening, everyone. Thanks for tuning in. I'm going to be talking about my PhD project, which finished up a bit over a year or so ago now, which was looking at better understanding orchid mycorrhizal fungi, with an eye on how that understanding can help a conservation of threatened orchids. I did my PhD up at ANU with Celeste Linder, but I was based most of the time at the Royal Botanic Gardens Victoria down at Cranbourne with Nushka Reiter on her orchid conservation program. So I thought I'd start with a bit of background on the orchids, and it's a bit of a hard sell to a group of mycologists I know, so I apologize for this. But orchids are plants, right? A huge family of plants, second largest family of plants in the world, with some 28,000 species or something like that. Um, and Australia's got about 1,500 species of orchids, so we're pretty rich. Um, and one of the larger genera is this genus called Prazophilum, or the leek orchids. So called because they don't look anything like a leek. But anyway, um, and all of those species are endemic to Australia. There's a couple of other species in New Zealand, but most of them are Australian species. And the center of diversity for leek orchids seems to be in sort of the southeast part of the country. And there's a particularly high number of species that occur in fertile soils or habitats on relatively fertile soils. And in the southeast of Australia, habitats on or vegetation communities on fertile soils are those that have just been broad scale cleared for agriculture in the past. And so there's a lot of threatened leek orchid species. Um, let's meet some of them, shall we, to put some faces to some names and give a bit of context for the background of this project. Um, I promise I'll talk about fungi in a second. Um, but you can see there's a couple of super rare species. Prazophilum gilgai from up near Benalla, only known from a single plant. Prazophilum correctum from near Bairnsdale out on the Gippsland Plain there, a couple of plants maybe, and a few more Prazophilum fosteri out west of Geelong. These species are on the absolute brink of extinction, and despite often the best attempts of land managers, the wild populations are headed down a one-way street. Um, and yeah, that's not a good thing if you're one of these species of lovely endemic leek orchids. Unfortunately, there's lots of other types of endangered orchids as well, rather than just leek orchids. And so much so that the Royal Botanic Gardens Victoria have this wonderful orchid conservation program that Nushka Reiter runs, where they grow these critically endangered orchids up in the lab. They plant them out into the nursery, which looks beautiful in spring. And then they plant them out into protected areas of bushland to create new populations of some of our most endangered orchids. Sorry, apologies for that. Um, and it works really well. It's the actually the largest endangered orchid rewilding program of its type in the whole world by like an order of magnitude. It's fantastic. Anushka won the inaugural Eureka Prize for botanical science research a couple of months ago, but because it's very successful, and they've brought a lot of these endangered orchids back from the brink of extinction. But they were never really able to work on leek orchids. For some reason, the seed of leek orchids just 9.9 times out of 10 didn't germinate. Now, this is where we'll bring in the fungi side of things. This is a fungi seminar. Um, the way orchids grow in the wild, so orchids have tiny seed. Like it's been a hot day in Melbourne today. You're probably gonna um, crack open the freezer after this talk and chow down on a bit of vanilla ice cream. And you know those little vanilla seeds in your vanilla ice cream. So they're the seeds of the vanilla orchid. Vanilla is a genus of orchids, and they're tiny, like dust. And all orchids have this tiny dust-like seed, and it's so small that the seed doesn't have any food in it, any endosperm, and it can't germinate by itself. So immediately you realize why there's so many endangered species of orchids, because they're stupid group of plants can't even germinate by themselves. Instead, they rely on their mycorrhizal, orchid mycorrhizal fungi, which are just sort of rummaging through the soil, stumble across an orchid seed, and instead of eating it, for some reason, they grow their hyphae into the seed and start to feed that seed nutrients and water and get it to germinate. And so to grow orchids for conservation purposes, we try and replicate this phenomenon in the lab. The fungi, orchid mycorrhizal fungi, usually hang on in the roots of the adult orchids. So we can go to the field, find an adult orchid, carefully take a little bit of root tissue, not too much because we don't want to hurt the orchid, but bring back a little piece of its root tissue, and inside the cells of the roots are all these balls of or pelotons of orchid mycorrhizal fungi. And we can pull those out, put them on some plates of agar, and they'll grow. You can see the fungus growing on the left-hand side of the plate here. And they'll grow across the plate. You sprinkle on the orchid seed, and what you usually find is something like this, where you see all these hundreds of orchid seeds in every dish just growing up like grass and pushing the lids off their petri dishes. But for some reason, that very rarely happened in Prazophilum, and that was a problem. So my PhD was sort of on trying to work out how to grow leek orchids, which is a totally plant conservation rationale. But pretty early on, we ruled out that there was any issues with like seed viability or anything like that. And we realized that it was, despite the project being a plant conservation project, it was really going to be a PhD on orchid mycorrhizal fungi. And if we could understand the fungi better, then we could work out why they weren't causing germination in the lab, why there was this breakdown occurring between the fungus and the orchid seed. So that's what I'm going to talk to you about tonight. I've got three main chapters, I suppose, that I'll talk about tonight, um, because we really did end up learning quite a lot about these orchid mycorrhizal fungi. So from the handful of small studies that have been done on the orchid mycorrhizal associations of Prazophilum of leek orchids in the past, we knew that they associated with this genus of fungi called Ceratobasidium. So Ceratobasidium are one of like the big three orchid genera in the world. They occur in orchids all over the world. Like, I don't know, if you go to Africa to do a safari, you want to see like your elephant and your lion and the that's a stupid analogy, I think. But anyway, they're a very common group of orchid mycorrhizal fungi that are found in all sorts of orchids, not just Prazophilum, but also in Terrastylus in Australia. They're the greenwood orchids. It's an even larger genus of orchids in Australia. And they also occur in some other strange orchids, Rhizoanthella. They're underground orchids that live totally underground and just produce a flower under the leaf litter, between the leaf litter and the surface of the soil. I've got a photo of it. It's bizarre things. And Ceratobasidium, also the fungi that associate with Sarcochilis. They're epiphytic orchids that grow on trees in rainforests up the east coast. Um, And you find Ceratobasidium in orchids, yeah, all over the world. First of all, I did a bit of a review chapter on what we knew about Ceratobasidium orchid mycorrhizal fungi in Australia. I was able to find over 500 RTS sequences from GenBank and also Big Trench of unpublished data. And I put them into a phylogeny with a bunch of named Ceratobasidium species because Ceratobasidium fungi are better known for being plant pathogens. They cause a lot of like rust diseases in wheat and other agricultural crops. Um, and when we put these known pathogenic fungi, they're the ones that have got a red dot after them. So these are all the different OTUs of Ceratobasidium. The one the OTUs that start with OTU-A, OTU-B, OTU-C, they're the orchid associating fungi. They get a little yellow dot if they've been shown to germinate the seed of orchids, so they're proven to be mycorrhizal. Um, and what we can see is that these orchid associating fungi are very closely related to, but different to the pathogenic fungi that found in on crops causing disease and that sort of thing, which is quite interesting because there was only one exception, this OTU-Q down the bottom here, which was found in orchids and was also apparently weakly pathogenic on strawberry plants in a study for some way, although judging by my strawberry patch outside, even I'm pathogenic on strawberry plants, so it's probably not yeah, some question marks over its the veracity of its pathogenicity. But we can see the orchids are associating um, with sort of the saprophytic end of the spectrum of this saprophyte pathogen spectrum that we know occurs in Ceratobasidium. Um, Although one of my study sites was at an aerodrome, and the guy who managed the aerodrome was the local farmer and um, not much of a greenie. And he'd always teach you about, oh, extinction's a good thing. If it wasn't for extinction, we'd have Tyrannosaurus rexes running around, to this very day. And what I should have said, if I had this data at the time, was that these orchids have worked out how to harness like the potential of a group of fungi that can be really nasty and very damaging, economically damaging agricultural diseases. Um, And I fantasize sometimes about there being some practical, hard, cold hard economic applications of better understanding how the orchids control infection in their root tissue by of fungi that, you know, can cause out of control infection or are closely related to other fungi that can cause out of control diseases in other plants. Anyway, I'm getting ahead of extrapolating a bit here. Um, what was also interesting is the diversity of Ceratobasidium orchid mycorrhizal fungi in orchids in Australia. So over the course of my PhD, I ended up with a rate of about one new Ceratobasidium OTU, totally different to anything on GenBank, for every two Prasophyllum host species that I sampled, leek orchid host species. Which, given that there's 140 something species of leek orchids, means that there's, a substantial um, number of unknown, undiscovered Ceratobasidium orchid mycorrhizal fungi out there. And some of these Ceratobasidium are only known from occurring in one species of really threatened orchid. So yeah, um, there's certainly a high diversity of Ceratobasidium orchid mycorrhizal fungi. As I said, some of them are quite rare and only found well, I'm not sure if the fungi are rare, but certainly they're only found in orchids that are rare. But other Ceratobasidium OTUs in this paper were much more common. And in fact, a couple of OTUs we found, we trawled through the CSI Rose Australian Microbiome Project database. So that's where that SIRO went out and sampled soils all across the country and just DNA sequenced everything that was in them, bacteria, fungi, and everything. And they've got a huge database with all that data in there. And we found that a couple of our Ceratobasidium motifs actually popped up with some relative frequency in that data set. And that meant that we were able to do some species distribution models of these orchid mycorrhizal fungi. Actually, the first species distribution models that have been published anywhere in the world, apparently, because um, this data, this soil sequencing data, meant that we had locations where the fungi occurred, presumably um, free living in the soil. They weren't occurring in the orchid roots, and that meant we could do some accent models and look at the distribution of these fungi. So this is one of the underground orchid species, Rosenthaler gardneri from southwest WA. You can see it, the mulch or leaf litter has been scratched away, and you can to reveal the flower underneath. Bizarre species of plant. Um, but the fungi that these really rare orchids associate with was actually modeled as occurring all over the country. So these orange dots are where the fungi was found in orchids. There's another underground orchid species. Well, there's actually two species over on the east coast. Um, the white dots are where the soil was sampled on the CSIRO Australian Microbiome Project, and the fungus was not recorded, and the red dots are where the fungus was recorded in soils. So you can see that the fungus, the Ceratobasidium OTC, that is associated with these underground orchids, actually occurs all around the country, up through central Australia, into the top end, and we modeled it as being particularly likely to occur in some of the wetter um, parts of the country. Um, and another species of Ceratobacillum that occurred with a critically endangered, this is that Prazophilum correctum from earlier in the presentation. It's actually used as a fungus that's really common across southern Australia. Um, the distributions of these Ceratobacillum fungi correlated with soil moisture and temperature variables. That's not particularly groundbreaking. I think the distribution of every fungus correlates with, or most fungi anyway, correlate with those variables. And certainly some Ceratobacillum orchid mycorrhizal fungi are very widespread. Others that were only found in like one species of endangered orchid and not found at all in the soils that were sampled in the Australian Microbiome Project may not be so widespread, but it's much harder to prove that. Yeah. Anyway, so we got a bit of a handle on some of the diversity and the distribution of Ceratobasidium orchid mycorrhizal fungi across the country. Then we wanted to dig down a bit deeper into, well, that's great, but what's happening actually in the orchids? What's happening in the communities of orchid mycorrhizal fungi that occur in adult plants and also in the germinating seedlings. So I picked 3 study species of orchids and sampled the roots of adult plants every 2 months over the course of a growing season, so from autumn to spring, because the orchids are dormant over summer. They just live like in a little bulb under the ground. And then in autumn, they merge, they send up their little leaf, and it gets bigger and bigger and bigger. And then in spring, they send up their flower spike and flower, set seed, and then shrivel back when it gets hot. So yeah, sampled the adults every 2 months, looked at cultures and direct sequencing of roots of the ITS, Barcode RTS region, and then compared what was happening in the adult plants to what's going on in the germinating seedlings. We wanted to know what fungi associate with seedlings naturally during germination. So I made these seed packets, which are like glorified tea bags, really, filled with orchid seed, buried them around the base of the adult orchids in autumn, watered them over the winter because it didn't rain, and then dug them up in spring. And some packets were full of these wonderful, beautiful germinating orchid seedlings. You can see them here. This is the start of the little leaf. This is it's sort of like a protocorm. We call orchid seeds that are this somewhere between a seed and a fully developing seedling, we call a protocorm. They sort of make this swollen bud thing. Um, and you can see them here in this is seed packet. This is a microscopic photo of of a germinating protocolm of Prazophilum lindleyanum, and you can see inside the cells in this protocolm are these little cotton wool ball coils, of pelotons of Ceratobasidium orchid mycorrhizal fungi. The older the fungi, they take on a more orangey color, and the newer ones are more of a clear, transparent color. Um, so then we looked at the mycorrhizal associations between adult roots and germinating protocoms. And this is what we found. This is a paper that's just been submitted to Annals of Botany. And we can see that our three leek orchid species are doing different things. Prazophilum frenchii had a very specific association in adult plants with just a single species of Ceratobasidium fungus. And in protocoms, it also had a very specific association with that fungus, but one protocom, which actually yielded quite a few sequences, associated with a second orchid mycorrhizal fungi, but all the others associated with the same fungus that was in the adult plants. So very specific association. In Prazophilum lindleyanum, that's a green leek orchid, it also had quite a specific association to two mycorrhizal fungi in the roots of adult plants that were frequently detected all throughout the season. But occasionally we would find a few others in the adult plants. But in germinating protocoms, germinating seedlings, they all associated with just one Ceratobasidium orchid mycorrhizal fungus, this green one here, OTUZ. And then Prazophilum validum was sort of somewhere in between, pretty specific to this purple mycorrhiza, but with a few others detected with some regularity. And the point we're making, well, there's two interesting points about this data. One is that understanding the change of mycorrhizal fungi communities throughout the year, or particularly between germinating seedlings and adult plants, is very important for conservation purposes because, well, it can be very important for conservation purposes. For example, in this orchid in the middle, Prazophilum lindleyanum, this green OTU, which is actually not the most dominant OTU in the adult plants, it's the second most common, but the more common one was this blue one. But this green OTU is the keystone Ceratobasidium fungi that this orchid requires for germination. And so if you're, for example, rewilding this species and planting out plants that you've grown in the lab to make a new population, then you need to grow them with this green fungus here. The blue fungi, the more common one, more dominant one in the adult plants, actually germinated the seed of this species of orchid in the lab, which is a bit tricky because it's possible that you could grow a whole population of orchids with this blue fungus in the lab and then plant them out into the wild, and they'll never reproduce if they don't have this green OTUZ with them. So that's a real keystone fungus that's critical in the life cycle of this orchid. So an understanding of this process is very important. The point we make in our paper that just been submitted to Annals more broadly is that do these orchid mycorrhizal fungi communities, are they changing through time? Well, in some orchids they're not, they're very specific. The orchids have a very specific association to this one fungus. But in other orchids, well, in the adult plants, the fungi communities do change through time, but it's only the presence or absence of these rarely detected OTUs that is driving that change. In orchids that otherwise have quite specific associations to one or two orchid mycorrhizal fungi. And the point we make is that studies of temporal change in orchid mycorrhizal fungi communities should focus on fungi that are likely to be important to the life cycle of their adult plants, which are fungi that are frequently detected and functionally significant, or that cause germination, support germination. Anyway, yeah, that's pretty much, I've said all of that. So that's very interesting background information to have on communities of orchid mycorrhizal fungi in some of our leek orchid species, but it didn't really help us work out how to grow these orchids in the lab, which was kind of the point of the PhD in the first place. So finally, we looked at the effect of growth media on germination in the petri dishes. So this is the slide from earlier on. When we try and grow these or germinate these seeds of these leek orchids, the fungus grows really well in culture, usually. But when we sprinkle on the seed, nothing happens. There's no germination most of the time. But we knew there's been quite a few studies that show that um, the composition of growth media affects the growth rate of different orchid mycorrhizal fungi. And so we were thinking, well, maybe there's something to do with the composition of this germination media that influences whether or not the fungus can or chooses to germinate uh, the seed of the orchids. Because I think orchid mycorrhizal fungi are sentient beings, and they definitely make decisions on their own accord, from my experience anyway. Um, so I focused on Prazophilum frenchii, using three different isolates. So this is that orchid that only associated with the one fungus, that orange fungus in the roots of adult plants. This one at the top here. So you can see Prazophilum frenchii only associates with a single OTU of serrated basidium orchid mycorrhizal fungi. I sampled three different plants, so actually quite far apart in the wild, and got three different isolates from those, one from each plant, sequenced them, and they actually had identical RTS sequences except for one or two ambiguities. So these are it's very similar uh, isolates of the same OTU. Tested them on 15 different nutrient media and looked at the growth habit of the fungus, sorry, and also the percentage germination. So first, this is a graph of the percentage of petri dishes, plates with aggressive aerial fungi hyphal growth. And you can see here that one of these three isolates, the isolates are in different colors here. One of these three, this green one in the middle, 8.2, on high macronutrient media, produced this really overabundant, aggressive aerial hyphal growth on almost 80% of petri dishes, whereas other isolates didn't. Um, so there was definitely some differences in the growth habit, or like the behavior of these three isolates that have got identical RTS sequences. And then when we looked at the percentage germination, we found this, which is very interesting and very exciting. So first of all, the composition of the nutrient media, these are all the different germination media along the bottom. The composition of the nutrient media is critical in determining whether or not these serrated basidium orchid mycorrhizal fungi germinate the seed of the orchids. This medium down here with the orange arrow is oatmeal agar, which was sort of like the industry standard nutrient medium that was everyone used for growing orchids symbiotically with their mycorrhizal fungi, and it worked really well for for a huge number of orchids. They all germinated just fine. But for Prazophilum frenchii, at least, we showed that this sort of industry standard, if growing endangered orchids could be considered an industry, this meter that we used for the last 30 years was totally useless for Prazophilum frenchii. Didn't support germination at all. And that confirmed, previous observations that these leek orchids don't germinate when we grow them or try to grow them on this oatmeal agar. But when we changed the composition of that medium and added either an insoluble carbon source in the form of wheat bran instead of oatmeal, which is a more soluble form of carbon, or we added macronutrients, then we did get germination. So the composition of the media is critical in 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 whether or not the orchid mycorrhizal fungi germinate the seed of the orchid. So which medium is best for germinating Prazophilum frenchia? Well, that depends on which isolate of fungi that we looked at, um, or that we used. So we can see this fungi isolate in the middle, this green one, 8.2. This was that really aggressive one from the previous slide, and it germinated, actually had the highest um, average germination rates of any fungal isolate on any medium on this high macronutrient medium without sucrose. But when we added sucrose, it hardly germinated anything at all. Whereas this blue medium, blue isolate 9.3 was pretty good on both. Um, and at the top, this red isolate 4.3 um, was not as good as the other two, perhaps, but germinated a reasonable number of seeds on low macronutrient media that had an insoluble wheat bran carbon source. So the um, yeah, these fungi that have got identical ITS sequences were totally physiologically and functionally different, and we called this the existence um, of strains of orchid mycorrhizal fungi. That, and it makes sense, I suppose, in a way, because some of these sites are underwater for some of the year. The other times of the year they're baking dry, and if they can associate with different, sub-OTU level strains of fungi that are perform better or under different environmental conditions, and that helps the orchids to persist, and that's kind of makes sense. But it does make you wonder what is the best like taxonomic level then to be asking questions about the relationship between orchid mycorrhizal fungi and their host orchids. Earlier on in the presentation, I showed you some maps of those orchid Ceratobasidium fungi that associated with the underground orchid, a really restricted orchid, but associates with a fungus that occurs all over the country. Well, the OTU of that fungus occurs all over the fungi, but maybe the orchid only associates with a certain number of strains within that broader OTU, and that looking at the OTU as a whole might misrepresent the actual going on. I don't know, but certainly when we see different fungi isolates that have identical RTS sequences that occur that um, are extremely different in their function and in their growth habit, then it makes you wonder what is going on inside the level of an OTU. There's certainly a lot of diversity among different individuals of fungi within an orchid mycorrhizal fungi species. Anyway, the other implication for this, of course, is interpreting results of germination testing, because usually we use germination testing to prove that an orchid mycorrhizal fungus is mycorrhizal and not just an endophyte that's hanging around and not, doing anything for the orchid. But these results show that certainly if you don't get germination, you need to take that result with a grain of salt, because it could have been that just the incorrect nutrient medium was used or something like that. More importantly, though, certainly from the point of view of the leek orchids, is that we did work out that we can germinate leek orchids symbiotically with their mycorrhizal fungi just by adjusting the composition of our nutrient medium. And we got, our germination rates for Prazophilum frenchia are only about 5%, but orchids produce so many thousands of seeds that's not a bad percentage germination rate. And they grew really well in the nursery and started flowering 18 months after germination. And it wasn't just Prazophilum frenchia, but a whole host of rare and endangered Prazophilum species were able to germinate and grow up, including some of our most critically endangered ones. It's a petri dish of Prazophilum fosteri that there's more little germinating seedlings in that dish than occur in the wild. And same for Brasiphyllum correctum in this pot. So for the first time, and now the nurseries at Cranbourne at the Orchid Conservation Program have got lots of Brasiphyllums, little leek orchids in them, and it's really for the first time there's uh, a real light at the end of the tunnel for some of these orchids that that had a pretty grim prognosis. And that's all down to understanding the role of orchid mycorrhizal fungi. So yeah, thank you for listening. Um, this project was supported by lots of people, including the former National Environmental Science Program Threatened Species Recovery Hub and the Herman Slade Foundation and the Victorian State Government. And I'd like to acknowledge the Aboriginal traditional owner groups whose land I did my fieldwork on. Thank you.
Great. Thank you very much, Mark. That was awesome. And yes, we do allow some plant talk at these seminars. So a reminder to those in the audience, if you have questions, please do pop your question into the Q&A panel and Mark will answer them. I'm going to kick it off with kind of a slightly sideways question. I was fascinated at the very beginning when you said, yeah, my PhD was to germinate that which really has never been done before. How do you approach a PhD where you've got such a high bar to clear? How did that come about?
Oh, well, we had some background intelligence. People had tried to do this before, and there'd been some, you know, people had germinated leek orchids very occasionally. Um, I make the estimate about 9.9 times out of 10 it didn't work, but it did occasionally work, often enough to give us an indication that it's not impossible. There's just something that's going wrong.
Hmm.
And the Orchid Conservation Program germinates a whole heap of other orchids, and there's nothing particularly special about leek orchids. They use this ceratobasidium fungus, but so do the terrastylus orchids, the greenhoods that that are easy to germinate. There's no troubles growing them. So there was no real uh, impossible issue that we could think of that would be preventing this from happening. It was just a matter of, going through the motions and doing the experiments, and eventually we're confident that even if we didn't succeed in the end, we'd definitely find out something interesting about orchid mycorrhizal fungi. But as it happened, we were were
Highly
on a
successful. No, that's great. A question from Mark, for you, Mark, from Tom May. Always great to see the combo of cute fungi and orchids. Have you ever isolated the orchid mycorrhizal fungus Ceratiobasidium from soil or other substrates? Do you think they have a stage that's not connected to the orchid?
I didn't do any soil sequencing in my PhD, although they were found. Some of the Ceratobacillus motifs were found pretty frequently in the Australian microbiome database. So that suggests that at least some Ceratobacillus species do occur, probably free living in the soil. Yeah, yeah, it's the conjecture. Orchids definitely need their fungus, right? So if they don't have their fungus, they can't germinate in the wild. Whether the fungi need the orchid is, it seems that for some fungi they probably or definitely don't need the orchid, but whether associating with an orchid confers some other advantage to some fungi, that's that's less clear. And there's been some recent research that's published that shows um, that maybe associating with an orchid gives a fungus a competitive advantage in the immediate environment around that orchid, because maybe they can shelter in the roots of the orchid, and from there they can go out and compete against other fungi that they otherwise wouldn't be able to. Yeah, it's that's the frontier of the science, I think, at the moment, and it's probably there's probably diversity of strategies. I suspect that some fungi do and some fungi don't, but yeah, it's difficult to prove. Yeah, I think that's
I'm
what
just going to have a
the
quick follow-up on that. So,
yeah.
going back to the idea of Serrated basidium being pathogenic on a number of hosts, if you look to see where you have your orchid distribution, do you have fewer of those hosts that Serrated basidium might be killing off?
Nah, that's I think that's a bit of a stretch.
Okay.
Because well, for a start, the ceratobacillium that are pathogenic are different species to the ones that are associating with the orchids. Um, yeah. Uh, yeah, I'm not sure. Yeah.
Okay.
It's probably yeah, it's an interesting question, but I don't think I've got the any can shed any more light on that with my
Okay.
data.
Another set of questions from the floor. Does the presence of certain species of fungi in the soil affect the ability for orchids to germinate in the wild? I assume they mean other than orchid mycorrhizal fungi. So that's the first question, followed up by, or with some of the more widespread orchid mycorrhizal fungi from your species distribution models, do you think that this would be less of a consideration compared to other threatening factors?
Yeah, so that back about 10, well, 10, 15 years, and that was a real hot topic in orchid mycorrhizal fungi. Yeah, back about 10 years ago was do orchid mycorrhizal fungi constrain the distribution of orchids? Do they cause rarity in orchids because the orchids associate with a rare fungus and then that's why the orchid is rare? I think maybe that's what the question is asking. Um, and well, there's no or very little evidence of that. There's been lots of studies that do those seed baiting experiments that I did. You bury a packet of seed, but not next to the adult orchids, but in places where there's no adult orchids. To see where these orchids germinate in the wild. And there's been a couple of these studies particularly in Southwest WA, and they've shown that these orchids can germinate in places where there's no adult orchids because the fungus is there.
Hmm.
Um, so yeah, I would say no, really, but with the slight disclaimer that studies that look at the distribution of an OTU, yeah, of the whole OTU might, at least theoretically, overrepresent the distribution of the particular strains within that OTU that associate with the orchid, that cause germination of that orchid seed. Possibly, that's yeah, a bit of an extrapolation from our results, but yeah, it's theoretically possible. Yeah.
Cool. Question from Marco. What is the definition of a strain to you? Is it a genetic variation within a species or an OTU? And are all isolates of fungi strains?
Yeah, so this is where I get myself into trouble for bringing in vernacular terminology that's not accepted by the scientific community. I mean, we use the term strain, yeah, loosely to talk about different lineages or something more than just different isolates within an OTU that that have different behavior. They're different to one another. They cause germination on one medium, but the other one causes germination on another. Um, I mean, there must be some genetic basis to this. And just looking at the barcode RTS region is probably not the best. Well, is definitely not the best way of determining whether isolates are genetically identical or not, because it's obviously that's not you can't do that. Um, so yeah, there obviously is some genetic difference with them, but it must be subtle. Otherwise, you'd expect to see some variation within that RTS barcode region. Are all isolates fungal strains? No, no. That's I don't think so. Um, Often we've isolated fungi from different plants that um that had also identical ITS sequences, and but those fungi didn't were not particularly different in culture. You know, they all did the same thing. They were all the same color. They all had grew across the surface of the agar, and they all, yeah, provoke germination to similar extents. Yeah, it's a bit of a gray area. Oh, yeah. That paper actually was published in Symbiosis, so in 2022. So you can I'll probably explain it better in the paper than I do off the top of my head. But yeah, there's
No,
obviously something on.
yeah, it was really interested to see that because I work with Physolithus, which is a mycorrhizal fungus of eucalyptus, and we see the same thing. We call them isolates, not strains. Now I'm rethinking that, but anyway, they have very different activity on host range, what they're using as different sources of nutrients. So it's very similar to what you're seeing. Um, and the genomic aspect actually might not be as subtle as you would think. Do you have any plans of sequencing the genomes of these fungal isolates? Do you still have, or strains, do you still have them?
Yeah, so all the fungi, or almost all of the fungi that I sampled in my PhD is stored um, at the fungi bank at the Royal Botanic Gardens, Victoria, down at Cranbourne.
Mhm.
Store them in little cryovials at -80, and they can regrow usually from those frozen samples. So yeah, they're all sitting there waiting for someone to play with them if you're interested. Um, yeah, I don't have any plans to do that. As a whole other That's someone else's PhD, I think. Um,
That's someone else's
but yeah,
career.
the fungi bank is a really cool thing, and there's a huge number of samples there from all the sort of the fungi research that Nushka and her other students are doing, and it's, yeah,
Hmm,
just sitting. Yeah.
cool. Okay, I have one last question if you're okay with that. Um, I really liked your musings about the fact of, what's happened in the orchid genome itself that allows it to potentially associate with this genera that is pathogenic. As you were mentioning earlier, they are different species. But have you ever thought about what it is about the orchid that might be allowing it to domesticate something that other plants have, die from?
Well, um, I mean, obviously, orchids have evolved over a really long period of time to control the infection by the orchid mycorrhizal fungus, and um, uh, they only control the infection of some fungi. You know, other fungi infect the orchids and then go on to devour them.
Mm,
Um, Not the orchid mycorrhizal fungi, but other species of Ceratobasidium. I did lots of germination testing, and there's only some OTUs that would cause the germination, and others would just eat the seed,
hmm,
or yeah. Um, so yeah, there's the fungi infect the tissue of the orchid, and then the orchid controls that infection, or the fungus limits the infection. I'm not sure which of those, depending on your point of view. Um, and that's how orchid mycorrhizal fungi work. And other plants are not good at controlling that infection by Ceratobacillum fungi, but they they are infected and have symbioses or mycorrhizal associations with other fungi, that
hmm.
including other fungi that are probably pathogenic on orchids. So it's not just orchids are not the best, although I think they are, but they're the best at controlling infection by orchid mycorrhizal fungi that are, yeah. Yeah, it's an interesting field. There's yeah, yeah, again, uh yep, that's for someone else who's more keen on that type of work than me to delve into.
Hmm.
But
No, very
but yeah,
good. Yeah, no, orchid mycorrhiza, they are definitely an area where we need to know a lot more. So thank you very much for taking the time to talk to us about your work today. Super interesting. As I said, thank you to those who had questions today. Thank you for taking the time to answer them. And we wish you all the best.
Thank you. Yeah, thanks for the opportunity.
Okay. And to everyone else, remember that this is a monthly event. So we will see you again next month, same day, the last Wednesday of the month, but we'll be back to our usual 12 o'clock slot. So thank you very much, everyone, and have a good rest of your day. Bye for now.